Hello and welcome to our coffee chat on Cell and Gene Therapies. I am Naima Mondral, Senior Editor of Bioanalysis Zone, and I'm delighted to be joined by two experts who have a keen interest in cell and gene therapies and their potential to change the way we fight disease. So firstly, could you introduce yourselves?
Hello, I'm Paul Byrne. I'm the Principal Scientist for Cell and Gene Therapy at Covance.
My name is Weiping Xia. I'm the VP for Biological Services at the Frontage Laboratories.
Thank you. So to kick off this coffee chat, let's start with the basics. What are cell and gene therapies and why are they important?
I think it's actually probably quite difficult to answer. I think if you go back maybe 5 or 10 years, it was a lot simpler. We had stem cells to treat like damaged tissues, or gene therapy, so modified viruses to deliver a therapeutic. But I think these days the molecules that exist under that cell and gene therapy umbrella are actually quite diverse and complex. We've got oncolytic viruses, we've got gene therapy and different ways of introducing those therapeutics. We have ways of modifying the DNA, regulating the gene expression. We have stem cells that are modified as well as just being embryonic or pluripotent. And finally, things like CAR T cells as well, these kind of immune modulating therapies, where the patient cells are actually modified and given back into the patient. So I think it's actually quite difficult to explain what cell and gene therapy is, although maybe I kind of try to do it there.
Yeah, so gene therapy has really changed a lot and evolved a lot. Back to probably several decades, first the gene therapy attempt, and then get to 90, and there was also a peak when there was a trial in the gene therapy. Unfortunately, there was inflammatory response. So then essentially kick the platform back to the laboratory to do further research. But now we see a great success in gene therapy, few products being approved. The key is how you get the gene into the body, essentially, right? Transport the gene into the body and incorporate and make functional. As Paul mentioned, there are various ways to do it and has been successful. I think in the next decades, you're going to see a much greater successful story impacting people's life.
Yeah, and it is relatively new as well, because again, there was only in the kind of 90s that was put into practice. And even then, the main headlines were actually failures. People died and developed things like leukemia as a result. So it's only in the last, I'd say maybe 5 or 10 years that things have really improved in terms of the safety and efficacy. And it's really interesting to see where we'll be in the next 5 or 10 years.
Yeah.
So you've touched upon how diverse they are. Can we now look at how these modalities have evolved and how did we get to this point in time?
It's a tremendous effort actually really put into, the industry is catching up, but really success comes from the laboratory, from academic research. And that's why a lot of collaborations between the academic and the industry is the key to the success of this particular emerging technology. UPenn is probably a good example, right? Work with, with Novartis and to get the CAR T out, right? So there's, of course, during the process, there's also setback, and people learn from that. And, back to the academic research in trying to understand better formulation, how to deliver, and how to control the safety issue, and how to make the transport gene more efficient. So it's just a new technology, new platform that it's so new to everyone. But it's so complex compared to, say, monoclonal antibody and protein therapeutics. So you have a lot to be concerned about it. And I remember even during my previous career, we're trying to get siRNA into the body, and the formulation becomes so critical. And you inject it into animals, and the animal get cytokine storm. Essentially, animal can be killed. Of course, you cannot get in a human, but people learn from that, how to control that, how to monitor that as well. So I think a lesson learned and also advance the research understanding this platform is very important. I don't think we are completely understand everything, and we're still trying to make this platform mature, even mature. But at the same time, there are new technologies, new platform coming into this play. Gene editing is a good example, CRISPR. Although we don't have a successful product in the market in this area, but a lot of companies are working in this space. I'm pretty sure we'll learn each other as well.
So it seems that cell and gene therapy, the industries are rapidly evolving, but more recently, I think, Paul, you touched upon it, how do these therapies fit into the bioanalytical space? Maybe thinking about the techniques and technologies that are evolving.
Yeah, I think, as I mentioned, the type of molecules that now exist within cell and gene therapy are quite wide and varied, and therefore the kind of bioanalytics have to evolve to accommodate that. Historically, we would use very sensitive molecular biology assays to look for the DNA, but that probably doesn't give us the answers that we're looking for some of these therapies, in particular things like stem cells and CAR-T. So I think from a bioanalytical perspective, the opportunities are in terms of developing those new techniques, or more kind of optimizing the existing techniques so that they can be more applicable to these different types of molecules and more sensitive.
Mm-hmm.
So this is actually, I think for bioanalytical it's a challenge, but also it's an opportunity as well. Because anything into the body, you have to know where they went, where they go, where they went, and how to monitor them. And this is really is in the bioanalytical arena. Traditionally, we use, for example, PCR to look at vector genome, essentially. But the PCR has some limitation too, because it has to do with sample preparation. There are some inhibitory proteins can be affected the PCR. So PCR not necessarily the best technology. Nowadays, just supporting gene therapy, there are so many new technologies coming. For example, digital PCR has been now even more popular platform in gene therapy. It doesn't need a standard curve to set up your assay. And also it can, because it's a droplet, it's a single PCR. So you don't have the issue with, for example, interference. So it's technically can be more sensitive. Although for PCR, you don't need sensitivity because you can always do amplification, but that technology is getting momentum and becomes popular.
I think it's a good point because, again, these new technologies are being developed, and I think people are still trying to understand where they're best used. Because digital PCR is very good for a number of reasons, but does it add anything within that preclinical or clinical space that you would get from the qPCR? There's obviously benefits to the digital PCR within the CMC, these advanced bioA techniques, to give you that much higher level of precision when titering these cell and gene therapy products. But I think that's one of the challenges, is just understanding what bioanalytical tests do you apply to your product, and then how you apply it. How do you validate it? So I think that's one of the big challenges.
The other challenge for the bioanalytical is there are a few guidance around gene therapy, but there is no single guidance about the new modality bioanalytical methods. There's a bioanalytical method for typical traditional PK, no biomarker coming for immunogenicity as well, but there's no a guidance really clearly define what is requirements, what expectations when you validate a PCR-based methods.
Yeah.
So there are some white papers based on their own lab experience to talk about, what's the best way to validate a PCR method, but nothing from guidance really to standardize.
Yeah, and I think it's a real problem
Yeah.
because I think a lot of companies, for qPCR in particular, just use their expertise to validate an assay and make sure it's fit for use. But everyone is approaching it in a slightly different way and probably generating data that's slightly different. So I think we do need some guidance. There might need to be some kind of education to the regulators to say, look, from a kind of industry perspective, this is what we're proposing to do with these assays, and get their buy-in, because I think for assays like mass spec, which we're starting to use for very small oligos, these interfering RNA molecules, they're very prescriptive validation requirements, almost too prescriptive. But then the regulators are used to that platform, the data that comes from that, from being used in the small molecule arena. So it's just a case of educating the regulators to say, look, it's not just mass spec now, it's qPCR. It could be flow cytometry for the CAR T cells. We could be doing other types of bioanalytical tests. So it's just having that dialogue, that education, so that they are quite clear what the expectations are.
So I think it's very clear that gene therapies are very complex and the methods and techniques used to evaluate them are developing. But you've mentioned some of the challenges, but what are the key hurdles for developing new cell and gene therapies?
I guess it's probably a difficult question for me to answer because I sit on the other side of the fence. So we support like the development. We don't do any manufacturing, but I think from what I hear is the actual manufacturing itself is one of the biggest challenges. So whether that's scale up, going from like a phase 1 to a phase 2 or 3 trial, for the gene therapy vectors, or whether it's for these kind of CAR T therapies, having the facilities and the resources to take these patient cells, basically modify them and put them back in. That's like a real challenge. Certainly moving forward, I think manufacturing is a big challenge at the moment.
So from our perspective, gene therapy, right now most popular AAV as a carrier, but AAV has pre-existing, human has pre-existing immunogenicity to AAV. So essentially, it's just pre-existing. So when you use AAV as a carrier, you make it neutralized and it becomes not effective, right? And I saw in the literature some AAV serotype can up to 30-40% patient who can neutralize AAV vectors.
Yeah.
So that's a really challenge. And the second is immunogenicity that we don't know what really means. That in addition to the impact on efficacy and what about the safety, right? So there are still inflammatory response that can be triggered, and so that's why people actually use immunosuppressant in combined with the AAV treatment, right? Just to control the inflammatory response, prevent cytokine storm. So those are the safety concerns, right? I think there are also a number of hurdles. One is regulatory. I think agencies are still expecting that when you have gene therapy, that the patient are mandated to be followed up for 15 years. I think I saw somewhere in the guidance, right? So the reason actually it's mandated to follow 15 years because really we don't have much experience with this type of treatment, especially gene, changing, addition, deletion, manipulation, this kind of. The second is ethical, right? So we're talking about a CRISPR gene editing now. So germline gene editing, where the ethics, coming from. That's what people are probably going to talk about, right? Yeah. And the third is financial as well. Technically, people just give one dose and they are done. So it can be expensive because it's not like traditional drug. You continue to take the drug and the manufacturer need to make it. Simply, people just get one injection, one treatment, they're done, So we're the financial support for the research and also from the patient and payer. Right, so these are these are the issues I think is even sooner or later needs to be discussed and figured out.
So you've mentioned some of the bioanalytical methods like qPCR, mass spec, and how they play an important role in ensuring the success of these novel therapies. You've mentioned some of the challenges, but what are the key bioanalytical challenges, and how do we support cell and gene therapies from a perspective?
Yeah, we've touched on just the diversity of the molecules and trying to find the best fit for those bioanalytical tools, and obviously how we then validate them to make sure they're fit for use. And I think we've also touched on the PK. The PK profiles for all these different therapies are very different. So it's trying to understand how we approach that within the preclinical and clinical environment as well. For example, these small oligos might have a PK profile of a couple of hours or days. The gene therapy vectors might be weeks and months. The CAR-Ts might be years. So it's quite tricky trying to understand what that PK profile looks like. And sometimes the preclinical data doesn't really give you any idea what to do in the clinic either. So sometimes you're having to readdress that challenge, even if you do have good preclinical data. I think the other consideration is just samples as well. For qPCR, mass spec to a certain extent, we can just freeze these samples and put them in the freezer for an extended period and then do the test. For the CAR-T, for the flow, for certain assays, they need to be done on fresh tissue. So just the logistics of how you manage that. Where is the testing done? Is it at the point of care? Are we shipping it to a laboratory with the potential impact that you might lose the kind of the material you're looking for? So yeah, there's a number of challenges there.
So talking about PCR, one of the sensitivity or low limit of detection people are talking about is a 50 copy per microgram genomic DNA, right? So I don't know where this number coming from, be honest with you, and what this number means, 50 copies per microgram genomic DNA.
It's actually hidden in an FDA clinical document that documents adverse effects.
Yeah.
So it's in a weird position, but it's the only, I think it's about 13 years old as well. So it goes back to the regulators in terms of that, educating them or getting more information from them, because all we have is in a very weird position in terms of the regulations, and is very old as well.
Yeah.
So.
And also, what about DNA low content DNA tissues, right? And what's the sensitivity should be there, right? So I think we still trying to understand what it really means in terms of some of the requirements, especially clinically meaningful.
Yeah.
What I see challenge for the bioanalytical is also, for example, immunogenicity. So in the typical immunogenicity, we talk about treatment emergent or treatment induced ADA, right, immunogenicity. And we use dilution factor, 1 to, for example, 1 to 3 dilutions. We basically say, okay, if it's threefold dilutions, then we call it treatment boosted. But for AAV type of gene therapy, the titer is so high, it's extremely high. It's not like threefold, it's way more than that. So what is the threshold that you can say this is actually treatment emergent ADA or immunogenicity, and this is not? So I don't think that the definition has been clearly defined, right? And second is for immunogenicity, we have to do like a pre-screen patient for the trial, right? And sometimes you run ADA and the ADA positive. But for ADA negative, sometimes you run the NAB assay and you get a NAB positive as well, right? So you could have like ADA negative, but NAB is positive, neutralizing ADA is positive. So you almost have to run both ADA and the neutralizing ADA to exclude patient in the trial, right? So you kind of limit the number of patients really can be put on the trial or can be benefit from the treatment. Right? The other thing is transgene protein. Right? So transgene protein, if the transgene protein is stay in the cell, well, it doesn't get exposed, so immunogenicity risk is probably low. However, if the transgene protein is a secrete, now you may get immunogenicity to that. And then if you have an endogenous transgene protein, there could be a risk. There could be a huge risk there, right? So these are the challenges, I think. And for analytical perspective, the reagent, any assay, especially ADA assay, immunogenicity assay, positive control. And how do you get positive control for those assays? And how you measure your sensitivity for your ADA assays? Those are a very important question to consider.
Yeah, and I think you're looking at probably like 6 to 9 months to be able to validate or get an assay up and running for immunogenicity. So that time frame has to be factored in as well. And I think we obviously speaking about the bioanalytical tests, which are kind of classically applied preclinical and clinical. But I think within that CMC environment, these kind of IND enabling assays actually covering quite a broad range of analytical techniques. There's the potential that we might address some problems there that have to then go back into the bioanalytical. Certainly for some of these new types of molecules that we're applying maybe a mass spec or a flow, we might get into that CMC environment where we're really interrogating the sample in terms of its purity, its concentration, its potency. We might actually find something out in those CMC assays that we then need to go back and readdress in the BioA. So we're just constantly finding out.
Yeah, so sometimes we have to combine different technology together to take advantage each technology, the unique. For example, gene therapy, we talk about, CAR-T, we talk about, AAV, but also, for example, endosense oligonucleotides, right, is also part of the kind of gene therapy as well. So endosense oligonucleotides, when you look at tissue, use mass spec, okay, you probably can do with the sensitivity. But if you look at the endosense oligonucleotides in the plasma or serum, it's challenging because you need really high sensitivity assays. And so we have developed what we call hybridization assays. Essentially, you use the DNA technology to hybridize to capture and detection, but also detect with typical immunoassay and trying to combine both to achieve the sensitivity you need.
Yeah, it's a really good example of what I was saying earlier on, having to try to optimize some of these methods to then support some of those BioA. And I think it seems to be the industry's changing a little bit in the sense that I think vendors would develop a new technology, then clients might take it on, or industry might take it on, and the regulators might become aware of it, and then people like ourselves in the CRO industry would then take it on. But I think now with these molecules and their very kind of like bespoke nature, we're almost actually innovating as well within the CRO industry because we're trying to basically answer questions where there's no kind of analytical kit for it. So yeah, I think we're all working towards new and novel ways of either pre-existing methods or we're trying to develop new techniques to basically answer those questions.
So if we move on from the challenges and now think about the success stories, what have been the success stories? And maybe you could give some examples of therapies that are currently on the market. What are we aiming towards?
I suppose the initial success was Glybera. So that was the first molecule to receive authorization approval in Europe. And I think that's when the floodgates opened, certainly in terms of us in the CRO industry, for people coming and asking for our services. But that ultimately has become a failure. It got taken off the market. I think they only ever sold one dose. I think each dose was at $1 million. So I think one patient in Germany was the only patient in the world to get it. Ironically, I think they actually did benefit from it. But I think just from an economic perspective, it just wasn't viable. But I think since then, they've improved safety and efficacy. There's quite a number of molecules that are now in the market. I think Strimvelis was a good example. It was developed by GSK. So that was a modified stem cell therapy to treat bubble boy syndrome. So they took a retroviral vector and then modified the patient's stem cells and then put in that stem cell that expressed the gene that was missing. And these patients went from having quite a difficult life, potentially quite short as well, to having fully functioning immune cells. So I think that dramatic kind of like cure is what I think is one of the big success stories.
Yeah, I mean, we're going to see more and more success story, right, from around CAR T, for the CD19, probably a good example in the leukemia, and also gene therapy as well. This year we approved, FDA approved for the pediatric muscular atrophy areas. So I think we're going to see a lot more success story.
Yeah.
And actually, we can learn a lot from that too. Even not just from the drug development perspective, not only from the platform itself, but even from the analytical perspective. I think we can learn from that as well. From, let's say, PK, look at distribution, look at the shedding, how to assess immunogenicity, what is clinically meaningful immunogenicity you have seen, and what it really means in terms of the benefits and the risk to the patient as well. Yeah. So analytical is supporting the drug development, really help to understand the exposure, the safety, but really the key to me is all this measurement has to be meaningful, has to really tell us what really matters to patient efficacy and the safety. Yeah. I think a platform, when I see this as getting mature and mature, and obviously continue to expand into the new territory, which we don't know much about it. But I think this is really set a good stage for us to go into, even in, as I said, gene editing. This is also a new area for the bioanalytical as well. We probably have to get into next generation sequencing, right? NGS methods to look at, when you do a gene editing, do you have off target, on target? You really look at the sequencing, the gene of the sequencing, right? So NGS, we don't talk too much about it, but I think in the next year or two, you're going to see a lot more.
Yeah. I think to come back to the successes, there's maybe only 2 or 3 products that have been approved onto the market in the last year or two. But I know the FDA are suggesting in the next kind of 4 or 5 years, they're expecting like maybe 20 approvals a year. So
lots coming through.
Lots coming through. And I think in the background, there's lots of new drugs showing amazing efficacy, whether they're first in man or later stage clinical trials, really kind of life-changing results. So, and I think once these companies have developed that vector, there's options just to flip out the cassette. So just to change the gene that they're treating. So I think once we start to see more of these products coming onto the market, it's going to be so much easier to manufacture, to do all the preclinical CMC and clinical. So things are going to ramp up quite quickly, I think.
So there are a few, actually not just few, there are many candidates in the development have been designated as a breakthrough. Mm-hmm. So this is you can see how promise in this areas and how FDA is pushing, right, give advantage breakthrough designation to speed up the approval process to get this promising treatment to the market and to the patient. Yeah. So you're going to see a lot more successful story in the next few years. Yeah.
So if we continue to look towards the future, what advances are we already seeing and what challenges do you expect to see?
I think we've touched on the analytics, just optimizing the existing analytical tools to support the wide range of molecules we could be looking at. So I think that's happening already. I can't see any maybe new technologies being invented to support. It's more just going to be a case of optimizing or potentially combining some of the analytical tests that we've got at the moment?
I mentioned before, gene editing is really, I'm looking forward to a successful, treatment from gene editing. And there are a few companies that work on in this particular space.
Yeah.
Gene deletion, gene editing, gene, insertion. It's going to be fascinating if we can successfully demonstrate.
Yeah. And I've been at the American and the European Society of Gene and Cell Therapy conferences this year.
Yeah.
And there's a huge focus on those gene editing. Again, a lot of the people presenting may be sitting in academia, but it's certainly a sign that it's the next big thing.
Yeah.
Yeah.
Yeah. And obviously, it's going to be challenging as well, right? Because as I said, this is you get to the line, perhaps more ethical issue coming as well.
Yeah.
So I'm pretty sure in the society will be a lot more discussion around that.
Yeah.
But this is really promising. I think it's going to be well received if there's a good story to tell and to demonstrate the benefit to really resolve issues people suffer
Yeah.
all the time.
Yeah. And again, you touched on the NGS.
Yeah.
I think that from a bioanalytical perspective is
Yeah.
a big challenge because they're easy assays to run, but they're very difficult to interpret the data. So the
It's-
bioinformatics side of it is
That's right.
a challenge because
That's right.
with these gene modifying therapies, it's not just checking that the correct modification has been made, it's making sure that there's no other off-site or target
off-target,
modifications.
yeah.
So and when you think about the size of the human genome and the amount of data that's involved in sequencing that, real challenges there, I think, moving forward.
Yeah, NGS is going to be interesting tool.
Yeah.
It's a very powerful tool, and but again, you don't have a really a clear regulation in terms of how you validate NGS assay. There's a approved diagnostic NGS kit assay, but that's only one. So a lot of even in the academic hospital setting, they trying to validate the NGS assays based on their knowledge and experience. Of course, the best practice, but there's no we call it standardized guidance around NGS at this point.
Yeah. And there probably won't be for a significant period of time. So
Yeah, people have to learn and then how to set the standard.
yeah, exactly.
So as the industry continues to develop, what do you expect to see in the next 5 to 10 years?
I think maybe two things. I think for the gene therapy vectors, obviously we've touched on the issues with like the immunogenicity, the existence of neutralizing antibodies and such. So I think they might maybe try and move away from those kind of viral-based vectors to something a bit more synthetic, that kind of almost bypasses that immune response, but also might improve tropism as well. Because a lot of these therapies, when administered, kind of go everywhere. So with the exception of ocular, so obviously it's, the ocular therapies are quite popular at the moment because you can deliver your therapy to the exact part of the eye that you want to treat. Yeah, but I think for the other one there is issues with kind of off-target distribution, especially into things like the germline. So yeah, I think if we can manufacture a kind of synthetic vector that allows better tropism, better efficacy, and less issues with immunogenicity, then I think that would be quite exciting. The other thing we touched on is the kind of things like the CAR-T, these personalised medicines, and the challenges with the manufacturing. It's kind of like maybe moving away from the kind of autologous nature of that to more kind of allogeneic. So there's more of a kind of off-the-shelf therapy, rather than having to have your own material taken away, isolated, grown up, modified, and then given back to you, which can take several weeks. So I think, and there is companies working towards that, but I'm not seeing any data to suggest how successful that, because obviously there's quite big hurdles to overcome in terms of that type of therapy.
What I see in the next few years from platform itself, I think it's going to be more mature, and people have learned a lot, and so I would expect in the future will be better efficient, in terms of transport gene into the body, and less, side effect like gene tox, and also inflammatory response. And from bioanalytic perspective, I think we're going to learn a lot from bioanalytical industry, and I would expect the agency would soon later come up with a guidance to standardize the practice for looking at vectors, genes, and transgenes, immunogenicity, including immunogenicity as well.
Yeah.
But also what I see on the technology front, especially for bioanalytical, there will be new technologies coming up.
Okay.
Right. And then to really support the platform evolution too. As I said, NGS will be, I'm pretty sure it's going to be getting popular. And also flow cytometry. And right now we don't have a guidance around flow assay either. So I think this is going to be, one of the next guidance will help the industry to standardize the best practice and support gene therapy and cell therapy.
And I think that'll all help bring these products to the market quicker, because I think that's one of the big challenges as well, is it can take 5 to 10 years to get these products into the market. So I think as we learn more about the therapies, as we can accelerate those preclinical and clinical packages, then I think the quicker they get to the market, the better, because they're not cheap to manufacture. So the quicker they can be made and onto the market, the more kind of economically viable they are.
Great. Well, I think that's a good place to close our coffee chat. I'd like to thank you for sharing your thoughts and knowledge today. I think we've only just started to uncover the potential of cell and gene therapies, and it'd be great to see how the bio community work together to bring more of them to market. So thank you.
Thank you all.
Cheers.
Cheers.
Cheers.